record on my computer, so you don't have to do anything. Okay, please continue. Sorry.
Okay.
Continue introducing yourself, and then you can start with your lecture, please.
Yeah, so I've been back to Thailand in Chulalongkorn University. This is my 4th year, and I have been working on biomaterials, especially the hydrogels derived from plants and extracellular matrix of the animals, and generally, yeah, because we think that it could be implied and applied to the tissue engineering, because you know, we create the microenvironment for the cells to grow. And I will talk focus on this topic in this lecture. So biomaterials, I made a lot of hydrogels. Now I shift a little bit toward bioprinting to create the bio ink. Okay, so yeah, that is for now. But yeah, let's take a look in the class topic today, tissue engineering. So I will walk you through the components of tissue engineering 101. And also, before going to that part, I want you to see this is what could be going on, and we can think about it. And when we talk about tissue engineering, so let me make a pointer here. So you will see the different color code, starting from blue, the scaffold, red, pink is the cells, and also the environment, which is the green one. So this is the 3 major components of tissue engineering. And I will touch a little bit of inside the component. For example, the cells, we have to know cell biology, different cell types, and also the scaffold. I will talk a little bit with bio ink and hydrogels and some scaffold that we create to let the cell stay inside the scaffold, and how to culture them with different aspects and different growth factors, chemical cues. Yeah, but last but not least, I will talk about bioreactor a little bit, since you have been studied before about mass transport, right? Mass transfer, yeah, in the previous class. Unfortunately, I will not talk about the top part, which is the orange zone, about ethic and more toward to the medical applications. Okay, so let's go back to times. Back then, people are very amazing because they think about to regrow and have a new organs when the organ get destroyed. So you will see the picture on the left side is the painting, and you see the guys who laying down, and there's a bird, which actually is an eagle, right? Yeah, so they try to take the liver of the guy. So actually, it's not the guy; he's a titan, and get punished from the Zeus because he steal the flame to human. So Zeus would send him to the Olympus mountain, and then punish him by let the crow to take the liver every day. But the good thing is this guy, Prometheus, he be able to regenerate the liver every day. So this is the concept of tissue regeneration. Amazing, right? Because yeah, people have this kind of thought all the time, immortal. And also on the right side, you will see the woman here. So she come out from the guy. The guy is what is his name? Adams. So, and then this is Eve. So basically Eve differentiate from Adam rib cage. So Eve is not like independent. So she's like something that come out from Adams. Yeah, so I want to show you this is the concept of regenerations and differentiation in the past, that people have this kind of thought all the time. A little bit further, so you will see the picture of the doctor try to implant the legs into the soldier. So back then they don't have idea, they don't have no idea is it gonna compatible or not, but they have this fantasies that we can save lives by take organs from other people and to heal the sick, the injured one. Okay, and this is the picture to show you that maybe we can control life in the confined environment. So this guy, what his name, Paracelsus. So he applies some chemicals, some substance in the vials, in the bottles, and hopefully it will recreate life. So this is a Renaissance era. So this is not new for us. So you will probably think, oh, this is a new technology, but actually they think before million years ago, 1,000 years ago. Okay, back to the current era. This is the definitions of tissue engineering. Tissue engineering is the interdisciplinary field that apply a lot of subjects, including engineering, applied science, but they have the same goal, which is to restore, maintain, and improve tissue functions and the whole organ. So basically we start from biology first. We know how the cells work. We know how the tissue functions and genetic materials. We know everything about biology. And also we have a technique to surgery and remove and reconstruct the malfunction shape of organs, like see the implant and prosthetics. But the engineering come later because human has a sense of control. How we going to make it stronger? How to make it more durable and can control the degradation over time. That's why tissue engineering incorporate biology and clinic together with the engineer part. It is very beautiful. And recently, you probably heard about the regenerative medicines, and apparently, tissue engineering. I show you by the laser beams here, come under the umbrella of regenerative medicines because tissue engineering turned to be one of the approaches how to regenerative medicine. Well, I don't know in the Thai language what can we use? ฟื้นฟูสภาวะเสื่อม let's say regenerative medicines. And yeah, there's so many approaches. For example, stem cells, use stem cell to fix the malfunction part, use tissue engineering to reconstruct, and also artificial organs. They are combined together to regenerate the loss of tissue functions and the whole organs. Why we need tissue engineering? Of course, because we want to restore the loss of tissues and organs, because most of the times our body part cannot regenerate after the disease or injuries. And yeah, we don't have enough the supply of organs and tissue. That's why we have to create by inside the lab and transplant it back to the patient. And also, when we create the new tissues in the lab, we be able to match the immune or the system of the organ that we culture and put it back to the owner of the cell. So there's no issue of transplant rejection because it's created from the original patients who have a disease. And also we prevent the disease transmissions. Sometimes when the donor, after they die, we have the system to screen the pathogens. If they have the HIV, hepatitis, ABC, yeah, of course we have that kind of screening. But sometimes that disease can transmit to the recipients. So when we regrow the tissue in the lab by using our own cells, we remove these issues. So this graph, there's a lot of going on, but I want to show you the line graph here. I got this data from Thai Red Cross societies in 2016, basically 5 years ago, but the trend pretty similar. So you will see the green line here, which is the number of patients who waiting for organ donation is in the 5,000 magnitudes here. But the bar graphs here, 200 and 500. 200 is the number of the donors. So we have only 200 donors who give the organ for 5,000 people. But actually we managed to transplant only 500 pieces, right, of the organs. What does it mean? Because some organs we have two set. For example, have 2 lungs, 2 kidneys. Yeah, so this is the major organ donation in Thailand. Yeah, and then I have to admit that tissue engineering it's not be able to create these major vital organ in the body yet. But of course, the structure organs we can do it. For example, skin, bones, I think cartilage as well. So the simple tissue we can do it, but the complex one we wait you guys to do it for us. Yeah, when the technologies come. Anyway, so you will get some impression that why tissue engineering is very important. Yeah, and the component of tissue engineering we can break down in 3 components, which are cells, scaffold, and signal. And I will walk you through each models one by one. Okay, and I think when I show you, and when we finish the cells part, we can take a break for 5 to 10 minutes if you want to, and then I will bring you to the scaffold and signal. Okay, so for the cells, I make a list here. You will see there's a lot of things going on here. For example, cells from primary cells. I use chondrocyte as an example, and then we can use adult stem cells. Now, everybody has the stem cells in your own body right now. And then I will show you back then, people are very interested in embryonic stem cells. And now we have another option, which is induced pluripotent stem cells as a cell source to create the tissue. And also the scaffold, I make a list of some of the example. For example, we have different type of scaffold from synthetic materials or the natural materials. Okay, and I'll focus on making the soft scaffold. And definitely the last part, which is the signal, which is the stimuli, what we apply to the culture system. And definitely, last but not least, we need time to grow, to recreate the new tissue. Yeah, so we put everything together and now we wait. Yeah. So, and also why it's important, because tissue engineering can improve the quality of life of the patient. I give you 2 examples of the patient who get the great benefits of tissue transplantation. So the first one, she's the patient who's suffering from tuberculosis. And yeah, so the doctor removed some trachea and recreate the new trachea and put it back to her body. And the second case, now I think she's well, almost 30 years old, and I think she still survived. But back then she had the problem with the bladder. So this is her bladder that recreated in the lab. So go to the first lady. So yeah, so she had the problem with the collapsed trachea, and you will see the trachea here is a long segment in the neck, inside your neck, and yeah, definitely they are internal and also have the cartilage ring wrapping around, because otherwise the tunnel will collapse. This is the structures of trachea, and what the doctors have been doing back then. Yeah, let me see. So it's happened in 2008. Wow, it's quite a big number. Yeah, back it's like almost 15 years ago. Yeah, what they have done is yeah, this is the condition of the patient. She have the complication of tuberculosis infection. So the doctor removed the segment of her trachea out. This is the patient. And how about the donor? The donor is a diseased person. So he's 15 years old and his brain is died, right? And then, yeah, so the doctor harvests the trachea of the dead donor, remove the cells out, and he reseed the patient's cells. So what's going on is in the right side of the slide. So inside here, you will see the kernel, and the doctors reseeded the epithelial cell, the cell from the patient trachea, the healthy one, like, so the segment is broken, but he take the above the bad one. Okay, so he get the healthy cells and reseed back into the kernel. And the second part is the patient cell, which is the bone marrow here. So the doctor see the cell at the outside of the kernel, and it's become the cartilage, the ring cartilage along the kernel. Yeah, so definitely everything will be inside the environment, which is the bioreactor, to give the nutrients, supplies, and it's actually spinning too, because it's make sure that the cell inside, they facing, or they experience the air and liquid interface. So yeah, so this is how engineers see and solve the problem. Oh, you don't have a trachea? Okay, we're gonna take the trachea from the dead one, and then we're gonna bring the cell from the healthy one and put things together inside the defined environment. Okay, so she's happy. And for the second case, which is the bladder malfunction, she has a congenital diseases. It's called spina bifida. What's going on is here. You will see at the back of the baby, and there's some spinal fluid exudes out. And what the spinal fluid does to her spinal cord is that it's compressed and it gives some pressure. So he lost the control of the lower part of the body, including the bladder. So she cannot empty her bladder, and cannot control how to stop and to, so there's some leaking problem going on here. What the doctor have been doing is that, so he create the engineer bladder. Yeah, so it start from the bladder of the patient, and then the doctor take the cells and culture the cells inside the lab, and then put the cells in the synthetic bladder scaffold out. Yeah, so we talk about it later. And also when it's mature, is this half sphere, if you can see carefully, it's not the entire ball. So it's just the top part of the bladder, and then surgery sew it back into the original bladder, existing bladder here. This is the new tissue. So when we take a look what's going on in each part of cells, scaffold, and the environment, you will see there's two types of cells that have been used in for bladder reconstruction, which is the endothelial cells, which is the cell inside the bladder, and also the muscle cell, because the bladder can have to be contracted and released, when you go to the toilet, right? So they need muscle to control the contraction. And yeah, so this is the cells part. For the biomaterials, yeah, so they use both natural material, which is collagen, and they have the second recipe, which is collagen combined with the polyglycolic acid. So they make like dome shape, you will see here at the bottom of the slide. And also when they put the cells and scaffold together, they grow into the culture media for 7 weeks to make sure that everything attached to each other, and take this to this operating room. Okay, so this is like expand versions of what I just said. Yeah, the cells have to take it out and then increase a lot of number of cells. That's why we have the word expansion. This is individual cells here, and then increase the number of the cells and put it back into the scaffold. So you will see the pink one represent the cells, and the thing look like fiber here is the match of collagen or polyglycolic acid. So yeah, the cell resides inside the scaffold, and when it's mature, they put into the operating room, and then the the new bladder integrate to the existing bladder. So this is what this look like from in the lab. Okay, so this is the overall. So we know in the past, in the imaginary of the people back then, and the benefit of tissue engineering and why we need this tissue engineering, right? And then we go to each component, cells, scaffold, and environment, the stimuli. So I will start with the cell first. Cell, everybody knows that is the building block of living things. And we have so many cells in the body, including, like from the brain cell top to toe. And it's carry the genetic information of each individuals. So there are 4 types of cells that have been using in tissue engineering: primary cells, adult stem cells, embryonic stem cells, and induced pluripotent stem cells. I will go one step at a time. So the primary cells, what are they? Yeah, so primary cell is the mature cell that isolate directly from patients. And if you remember, we just said about bladder, tissue engineering bladder. So we take the cell directly from the mature person, from the patient own cells, and then we expand and do everything and put it back. So definitely there's no immune rejection. Yeah, so this is primary cell. You probably heard about they take the cells from the skin, they take the cell from the knee, something like that. So it's from the original source. Yeah, so when you take a look at these pictures again, so this is the primary cell, I would say, here. So the healthy part of the trachea. Yeah, but for the right side, it's taken from the patient cell, but it's called adult stem cell because the bone marrow, like a red part in your bone, there's a lot of blood and stem cells, so they call this is adult stem cells. And we will talk in the second sort of the cells. But I want you to focus on this one first, because when we take the cell from the patient directly, we see it back into the scaffold right away. But this one, we have to make them transform. We use the word differentiation. You maybe heard about it when you read the paper. So it's not the cartilage at the beginning, it's something else. It's the stem cell, and then we transform them to be other cell types. Yeah, so the primary cell is just this one from this project, from this tissue engineer trachea. Yep. And so the second part that I just touched a little bit, which is adult stem cell. So I make an underline here. So stem is mean it's like a root. Yeah, it can become other things, rather than themselves. And that's why when you heard about stem cell, what are the characteristics of stem cell? Basically, there are 2 things, which are they can renew themselves, they can expand and divide it and become themselves to have a lot of cell number. And the second characteristic of stem cells are they can differentiate it, which is can become other cell types. Yeah, so you will see embryonic stem cell, even adult stem cell, they share these 2 characteristics. Yeah, and then when they become other cell types, they can repair the loss of function of that tissue. So I show you this one. This is an example. They are the stem cells from bone marrow. When you zoom in, there are so many blood types in here, and the stem cells in there can be replicates, dividing to be their types and become other cell types as well. So I show you here. This is mesenchymal cells here. So as I just said, they can replicate, which is self-renewal. They become themselves, and also can differentiate. become other cell type, for example, epithelial cell, neuron cell. But what we're gonna focus to make the engineer trait here, this is the one, cartilage cells, which have been seeding outside of the tubes here. Yeah, they take from the adult stem cell from bone marrow of that lady, that patient, and trick them, stimulate them to become the chondrocyte. Okay, and not only limit into the bone marrow, there are some stem cells in the fat tissue as well. So we call adipose derived stem cells. And other part of the body of adults, the one that come out from the belly of the mother, right? So this is adults. And there's some stem cells in umbilical cord, side, yeah, so they have some kind of business that collect the umbilical cord of the babies. And surprisingly, our nervous system still have stem cell, but it's very rare when we get older. Yes, skin stem cell, I think, yeah, definitely we have it. Well, how we know it? So when we get the wound and injury, so our skin can replace the old one, the dead one is very quick. And also when we have some scar and everything, so that means the underneath skin can replenish the top one that lost. So with anything that can fix, that means they have the stem cell underlining. Okay. Wow, that's a lot of things here. But yeah, we can start from the bottom one because there's two cells types I just talking about. The first one is primary cell we take from the adults or mature people. So it's already defined the function. For example, we take it from the bladder of the patient, also from bone cartilage. Yeah, this is the terminally differentiated cell. They define the function already, and we take that cell to recreate the new tissue or organs. The second layer of the cell that I just talking about is adult stem cell from bone marrow. Yeah, and you will see this is I draw the arrow which can cell renewal and become other cell type. But this one, they cannot do a lot of cell renewal because it's already old and already aged. They terminally differentiated. But this one, there still have some room to differentiate it. For example, the cell in ectoderm, which is the skin and neuron, and they can replenish themselves. Endoderm, actually, I don't know if you, maybe, I don't know if you still have the biology class 101. Yeah, so because we have three germ layer, endoderm, mesoderm, and ectoderm. So endoderm responsible for internal organ, for example, pancreas and lung, and they can differentiate themselves and become lungs and pancreas cell as well. So we just talk about only two sources of the cells. And now we're gonna move to the further step, the number 3, which is embryonic stem cells. I have the H here is human embryonic stem cells. So they have a lot of potency because they can become themselves and can differentiate to 3 different germ layers. Very powerful than the second one. Yeah, the third one is very powerful. So this is the Professor James Thompson's. He is the one who developed human embryonic stem cell in the standard way. And yeah, so what I written here, of course, yeah, so they have prolonged undifferentiated proliferation. They can proliferate and divide themselves, cell renewal, and also they can develop to other 3 germ layers. Yeah, differentiation, definitely check and check. So this becomes stem cell renewal and become other cell types. The embryonic stem cells, where are they from? So they are from these steps. So it's called blastocyst, but not the entire balls, just the inside, the blue tiny little ones here. Is called inner cell mass. So his lab take the inner cell mass and culture in the petri dish in the lab. So this one can become other cell type, because when he collect them and inject it into the leg of the mouse, yeah, the rare legs of the mouse underneath the muscle. So you will see here, but when he culture or leave it for 7 to 8 weeks after injection, there's some nodules formation under the skin. And when he make a dissect and take a look under the microscope, it's a little bit scary, because since it's very powerful, the stem cell not decide which one they want to be. They become everything. For example, this one look like a lumen in the intestines, and this one look like a bone cell, and this one look like a cartilage. So there's a mixture of tissue inside the ball. This one is look scary. So we call the balls teratoma. And I have heard that you probably see in the internet when they make a section and cut in through the teratoma, maybe there's some hairs growing. So yeah, so when you be careful with inject the stem cell, I think this is the consequence or some warning red flags that you have to be considered and carefully, that maybe it's not good. Even it's very powerful, but there are some side effect, which is not quite impressed. It's pretty much the same information I show you here. When they make a sections, you see a mixture of the tissue. Look like a intestine, look like the fat cells, look like the muscle cells here inside that balls. All right, so we just touched the third part of the cell source. You will see there's some strength or potency. This is, we call it potency of differentiation. Very low at the bottom, a little bit higher, and higher here at the top. So there's another vocabulary here, undifferentiated. They not decide what they want to be yet. So but this one is already differentiated or defined. Okay, and the fourth part, the fourth cell types in tissue engineering, we call induced pluripotent stem cells, or IPS. This one have this original source from the primary cell. I would say go back to the first one. In this case, they use the skin cells of the patient, but the scientists reprogram to make the skin cells become stem cells like. And then so we'll see there is somatic cells, or the cells from the body, from skin, from nerve, which is already define the function, but they want to make them to be the stem cells here. What they do is they apply the star, the red star, which I expand into the bottom part of the slide. They call transcription factor. These are the factors that reprogram to make the skin cell forget what they are before, and then become the stem cell here. And then we back to the original, and then we differentiate, make them to be other cell types, like start from skin, and then the skin can become the nerve cells, become the muscle cells, because they forget what they used to be. And then this one, the specialized differentiated cells, they can go back and treat this patient when he gets sick, right? But this one have other application as well, A, B, C, D. And the tissue engineering part that we probably involve the most is the D, and I will focus and talk a lot about this more. But I want you to know that they have another potential as well. When we have the pools of stem cells, and then we can generate the disease models of this patient. Is very customized and precise. Precision medicine is come from here, and also drug screening, because sometimes one patient is not respond to the drug. They need some different dose or different medication, and then we can use her or his cells to screen the effective drug. It's very specific to herself. And also we can use these cells to test for the toxicity. So it's kind of, that's why we have the word called specialized medicine or precision medicines, because it's specific to the original patients here. Screening, modeling, and test the toxicity. And yes, the D application, which I just said, it's can be the benefit, can give the benefit to tissue engineering, because we can take the primary cells from the skin of that patient coming to the lab. So these are the skin cells, they still remember, and then we transfect the transcription factor, we call the word reprogram, into induced pluripotent stem cell, and you see there from the colony exactly look like the stem cell, which I go back here. So you will see here, so it's become the group of the cells. So it's reprogrammed to be a tiny little colony. And then there are two options in this step. So we can freeze the cells in the liquid nitrogen to preserve. It's like a spare part of the body. So we keep it here. And some of the cells here can go for therapeutic. For example, if this is for cardiac cell therapy, so they make the stem cells here become the cardiac cells. And so it's called cardiac progenitor cells. So it's going to be the part of the heart of the person. And so they differentiate to trick them to be three different cell types. For example, the cardiomyocytes. But the cardiomyocytes cannot stay alone. They need smooth muscle to coexist. Not only smooth muscle, they also need the epithelial cell. So they combine these three cells together and seed it into the scaffold to make the engineer cardiac patch here. And then after they get the patch, they transfer and put it back into the heart to fix the infarct, like เลือดขาด หัวใจขาดเลือด. They said that. So this is the potential or the application what tissue engineer can serve the community, right? So this is the full function of tissue engineer. They have cells, they have scaffold, and also culture in the defined environment to get the mature one to put it back. And you probably see the different arrows. For example, here, when after they get three different cell types, they can inject back into the veins, the blood vessels here, and it's gonna be into the heart. So they let the cell do it by themselves. So this one, we a little bit engineer them together, but here let them flow into the vein. And another part is inject into the heart directly. This one go through the blood, this one go to the heart. It looks like a sci-fi. I don't know it's happening yet, but to give you some idea that the tissue engineering and stem cells could be the possible treatment options in the future. All right, so I'm pausing from now. How do you have any questions or some concern and some ideas to this part of the talk yet?
Ask a lot of questions. Or you're afraid of cells? Anyone want to ask anything? Or we're gonna do a quick break, right, Ajahn?
If you want to. So
okay. Normally we do 15-minute break if we can. What time is it?
15. 45.
Quite early, actually.
Quite early. Okay, so it's up to you. I'm happy because I'm just make sure that everybody's happy there.
we can pause for questions. If you have any questions, ask now, because you actually have to do the summary. Can you summarize for us again about the cells that people have been using for tissue engineering, and which cell have the most potential at the moment?
Yeah, because right. Actually, let's talk this way, because I forget to mention the drawback of each cell. For example, the first one, the primary cell, is plenty, but when we take them out and culture them into the lab, they have what is called they cannot divide or expand forever because it's already aged, right? It's aging. This one, because for example, if you are 25 years old, you take the cell, you're 25 into the lab, and they keep expand, dividing. They already the starting point is 25, and also it's a little bit this one come from your body as well, but it's the stem, so they have more chance to develop and divide and become other cell types. So basically, number 2 has more potential in terms of become other cell types in the tissue more than the first one. So this one is kind of like stop. This one, okay, I become myself and I become other cell types. And the third one, I barely heard about the third one research, because I think they have a lot of issues involving with the embryonic stem cell, which come from the freezer. Yeah, IVF, the in vitro fertilization clinic. And yeah, so people say, oh, when life starts, if we destroy the embryonic stem cells, this is called killing the babies or not. So I haven't heard much about embryonic stem cell. If you ไม่ค่อยได้ใช้แล้วเหมือนกับมันเป็นจริยธรรม ethic involved with this one a lot. And also this is not come from you, it's not from you. When you transplant it back to your body, you can reject. Yes, go back to the very first concept why we prefer to culture our own cells and tissue, because it's us at the beginning, but this one is from someone else. And number 4, it come from the patient as well. Yeah, so I just show you the skin and become the heart cell by using this one. So this is what I just think about and add on for now. Yeah.
Okay, thank you. So in terms of viabilities and in terms of safety and law, so they have their own good point and disadvantage, advantage and disadvantage. But the one that used actually right now for the cell therapy in human can be primary cells or adult stem cell mostly. Is it?
Bone marrow.
Is IPS on the real use already or not?
I heard about that too, but the trigger part of IPS is during the reprogramming, because we use the genetic engineering and make sure that they can convert the skin to become the stem cell first and differentiate. So there are many steps, and have to make sure that each step passes.
So you got the stem cell, you actually manipulate stem cell a lot in the lab before using it for the IPS. And the reason that IPS get very famous because they happen to be some Nobel laureate, right? The scientists who get wrote a Nobel Prize on this one, but it's just a potential at the moment. It's not really on the clinical trial yet. Only primary cells and adult stem cell, which is have been using a lot at the moment for the reality. Okay, thank you very much, Ajahn. So I think let's go ahead and try to stop for a break about 2:30. Or before 2:30. I think you can provide, you can divide it by yourself. Thank you so much ค่ะ
ต่อเลยไหมคะหรือว่าพักกัน
ต่อเลยค่ะเพราะว่าจะหยุดประมาณบ่าย 2:30
โอเคได้ค่ะ Okay, so we go to the first one, which is cells, and now we go to the second one, which is scaffold. Scaffold, I just take it from dictionary. Definitely, it's not forever. It's temporarily. Yeah, it stay for a while. So this is scaffold. It's a structure outside the building. This is what you heard, and it's the word that people using a lot now. But in biology, it's mean like a place for cell to reside in. Okay, let's read together. The temporary structure outside the building, made usually for wood, planks, and metal poles. Yeah, so it's basically it looks like this. This is a scaffold everyday life, and people will see it. But when the construction finished, the scaffold will take it out. The same thing as scaffold in tissue engineering. We let the cell to fill the gap, to fill the holes here, and then the cell will secrete the extracellular matrix themselves to lay down, and then the scaffold will degrade over time, right? And when you see it here, just focus on the cell, which, for example, this the blue tiny little one here, the yellow one here, and also the pink one here, the purple ones here. These are the cells, and they are surrounded by a fibrous structure. For example, the yellow one could be collagen, and this could be proteoglycans, or the matrix we call extracellular matrix is outside the cells. Yep. So the name is just when we zoom in, this is the cell membrane, right? And this is the intracells here. And the extracellular matrix is the part on the top or outside the cells, composed of collagen, which is the purple bundles here, and some proteoglycan, which is the blue here. Yeah, so in tissue engineering, we try to construct or create the extracellular matrix, the environments for the cell to get into it. Yeah, there are many approaches. Yeah, so I can show you there's some, for example, here, the first one, what it look like, we call pre-made porous scaffold. We have the different geometries here, the cylindrical shape, the cube shapes, or the rod shapes. And when you take a look inside, zoom in, yeah, there's some porous scaffold, and they have different approaches to create the geometry like this. And then you see the pores. These tiny little pores is where the cell can get into it. So they have the graphic here. The cell is tiny little sphere here. They fill inside the pore. So we call cell seeded. They're already seeding inside the porous scaffold. And then when we culture them in the lab and ready to implant into the defect tissues. So for example, this is the first one. Go to the fourth one, is pretty much the same idea. So we have used hydrogel. Yeah, so we encapsulate, which is mean mixing the cells into the hydrogel material. Hydrogel is look like a jelly when you have the pudding thing. So it's very soft at the beginning, and you will see tiny little thing, the cell inside the jelly. And yeah, and then we mixing together, we culture in the lab, and then we can make it grow tough and firm like a cartilage and put it back into the body. Or you not make it very stiff one. We make it like a liquid, like a toothpaste, and can inject into the knee, like the defect here, something like that. So this is the number one and number four, which is very easy. And it's like a classical technique for tissue engineering. Number two is pretty interesting, and we are in our lab are focusing on it, because decellularize extracellular matrix. It comes from the tissue, and then we wash out the cells from the tissue. So basically, if you remember in the very first slide about the trachea, so let's say this is a trachea, and then we remove the cells of the donors. So we get the decellularization, remove cells from the original tissue. And then there are some pores in here, and then we put the cells of the patients inside, and then we implant back. So this one very simple, direct forward. Yeah, this is the second. The third one is actually using scaffold, but it's very interesting, and I want to show you. So they grow the cells into the tiny little sheet, and then they secrete extracellular matrix and make it look like a tiny little patch. So they stack them together. So in each individual sheet, they stack together and it's thicker. So it can be they always use in the cardiac tissue engineering. So they stack the tiny little sheet, individual sheet, into the thick one and implant it back to the patients. So but now they have so many things to many approach, many method to create the scaffold. Yeah, so this is the visualization that I want to show you. So start from the simple one, the skin, like this. So when the cells go out, you will see some pore. I think it's the hair cells, the hair follicles back then. And we can use this for wound healing at the skin. And bone is pretty simple forward as well. So you will see some porous at the bone tissue here, but before that it's gonna be red because there's bone marrow, and the patient, the surgeon, they use this bone a lot, like we call bone graft or these like bone matrix to fill the gaps of the after the surgery. These are like, okay, this cornea as well, they vary the different type of detergent. This is a native cornea, look like this, and then they use Trident X and use SDS, different detergent, and then use another detergent plus at the cold temperature. And this is the outcome. Back then, the native cornea, which is in the eye, there are some cells inside, and after they remove cell, yeah, there's not many cells in there. This one is not effective, but compared to the SDS, another detergent, you will see there are some pores inside, and there's not cell deposit into the matrix. So when they improve with different detergents and incorporate the temperature in there, so from the native tissue, the native cornea, there's no cells in these two approach. So I think it's kind of like a shortcut, so we don't have to create the scaffold and like everything to at the beginning. We take what the tissue have, just remove the cells out, and we use the scaffold and the metric from the owner, the donor. So, and then there's some experiments. So they try to remove the cell out of the heart before watching and after the watch, liver as well than here. So we don't have to. It's very very difficult to recapitulate nature. So they make a shortcut, rather than they make a tiny little lungs. The balls here, we use the lung from, this is the rat organs you see here, the kidney as well. It's pretty impressive. It's beautiful. And we try to play God, basically tissue engineer. We try to make things that's similar to the original one. All right. So I would say I am from the classical tissue engineering because it's like 10 or 20 years ago, the technique to reconstruct the 3D scaffold. We start from what we want to make, which is, let's say, the block like this, and we create the holes inside, the porous inside, and then we put the cell into that scaffold, and then we culture them into the defined environment that I mentioned repeatedly. And then when it mature, we got the engineered tissue, and maybe when we put into the operating room, it's actually sphere, so we trim it out, like from the block, and the doctor need the sphere one. So it's kind of very hard to control from the classical perspective of tissue engineering. And then we have the bottom up approach. So let's say this is the final thing, the final outcome. I need the block of tissue like this. So let's play like a Lego, unit by unit. So they make a tiny little square first, and then we combine together into the bigger blocks, for example. And also we print it. I will show you here a tiny little layer by layer, and then we combine them cell assembly to become the big one like this. So the final thing, the final outcome, pretty much the same, but the approach are different. And now I think in your generation, people prefer to use the bottom up approach, the tiny little units, and make a big things. Yeah, and then if we don't want to make a block like this, we want to make a sphere, we can design the process at the beginning right away. We computer program it to pipette or make a dot, tiny little dot into the big sphere. Yeah, so from this approach, we need to subtract or use some art and craft to make it like a ball from this block. Okay, and this is the picture from my lab. We try to develop the bio ink for printing the tissue. We use two different polymer. The first one from hyaluronic acid, the one that the cosmetic surgeon use for make a filler, the hyaluronic acid, and we combine with the seaweed polymer, which is alginate. We modify the chemical functional group, so when they separate, they're not become the gel yet, and then we combine together. They develop the covalent bond and become the hydrogels, and then we vary the ratio. Hyaluronic acid 7 part and alginate 3 parts. It look like this, but it's not that firm. It become gel, but the printability is kind of not precise. But when we have 1-to-1 ratio, hyaluronic acid 1 part and alginate 1 part, it's pretty good. When we print it, it's controlled. The fluidity, viscosity is very good. Yeah, and this is some example that we make the ink from the HA and alginate. Yeah, we try to go to that direction. And this is the custom made. It's like a garage. Custom made 3D bio printer. The name is Enterprise 2.0. We have the 1.0 before, it looked ugly, and then I demolished them, and then we create the new one, which have like box like this. They have the UV light for sterilization and have the laminar flow HEPA filter on top, and be able to print in a defined string. So you will see it look like this, and now we get better and better. So we make a tiny little one, and we want to make the ear shapes of the human body. Is you will see here, but actually it's very tiny, around 5 centimeters. So it's like I just want to prove the concept if we can print it. Yeah, so it's used. Yeah, we made it in the lab. Okay, so scaffold. I'm not go to the details. I don't know if you I don't know, Ajahn Soroda, that the student take class about
Biomaterials.
and biomaterial before.
Okay. Yes.
Yeah.
But you can just kind of make a summary that what kind of target of designing scaffold you want to design scaffold to be what, to do I mean to have some the very important characteristic of scaffold. Actually, what kind of thing you want to control? For example, the shape, degradation, or mechanical properties, or something like that. Before we take a break, I think just summarize, what important in scaffolding. Please.
It's very good. Yeah, that Ajahn Soroda mentioned this, because as we are engineer, we kind of don't have the impression for the tissue function. So basically, when we want to make any kind of tissue, we have to think function before form. It depends. So sometimes when you have a different school, make a form and then it become the function. So, but here, if we want to make anything that serve the purpose of healing or treat the patient, we have to think, this is cartilage, or the function is support, and for shock absorber, that means that your scaffold has to be resistant and tough enough, not very fragile, something like that. So we think about function first, and then we step back to think about the properties of the materials and the type of material we fabricate to become the cartilage of the bone. Yeah, this is good. Yeah.
Okay. Again, we have to pause for students to ask questions. Anyone want to ask question, please? Or you want us to kind of explain something and you cannot catch it? It's too fast for you.
Can you explain about the bottom-up approach again? ครับ
Bottom up. Okay, ได้ค่ะ so bottom up, it means we use the subunit of combine together, the subunit to combine together, like could be individual cells and print it. A little bit bigger here is called cell aggregate. So this is one cells, and then we make it by printing. And here we let the cell aggregate, let's say in each tiny little ball has 10 cells, and put it on the side first. Or bring the cell into the tiny little hydrogel, make a block of 10 cells. And for example, here you will see the example. We make the sheet of 10 cells. I just put random number 10 cells. So this is the Lego, the subunit. And then we combine them together. For example, the cell assembly or the cell sheet here, they're stacking up until they got the big size like this. So this is the from bottom tiny little one, make it bigger. Did you see that is clear? More clear?
Yes ครับ โอเคครับ ขอบคุณครับ
More questions on this? I think you might be more comfortable with talking about scaffold because you have a lot of class on materials already,
Yeah.
right?
That's why I didn't prepare into the detail how to
Okay.
or freeze drying. I think you probably been through all these things.
But if you said, how do I know that I want the scaffold to stay for some time, but how do I know how long my scaffold have to stay to make this tissue? In order to do this, what kind of data I have to search on? Do you have comments on this, Ajahn?
I think.
Is it too hard?
Yeah,
Even I don't know. Don't know the answer of this.
Other tissue, I'm not sure, but for cartilage, it will take around 2 or 3 months to fill the gap. You see the holes here. Okay. And then the cell that we encapsulate inside the pores. So if they secrete the matrix faster, it's good. Yeah, and then the degradation term is for the research. You make sure the porous scaffold here will disappear over time, but have to make sure that the cell will create their own materials, their own extracellular matrix right before the degradation of the scaffold. It's like an arc, basically. Art and science combined together. Yeah. We define the shape, and then we measure the degradation rates.
Okay. All right.
Yeah.
If there are no more questions on this, I think we take a break and we might come back around 2:20 to continue. So you finish all the scaffold already, right?
Yeah.
And then we will talk about signals, which is the third component. Right. Okay. So everyone take a break, please, and come back around I think 2:30. That's okay. 2:30. Yes. Yep. Thank you, Ajahn. You can take a break too.
I'll see you later. Sure.
Okay.
Sounds good. Let me share the screens where we were. Go. Face.
Oh, worry.
Okay, my face is here. My screen. I already shared the screen, right? Okay. Good. Okay. So the last part of the tissue engineering component is now here. Okay, so tissue engineering triad, we talk about cells, scaffold, and now we're gonna move to environment or stimuli. So I make the green components here. So it could be chemical cues, cytokine, extracellular matrix, mechanical loading, or anything that we applied in the cell culture system in the bioreactors. Yeah, so we put the cell, combine them with the scaffold already, and now how we gonna culture them, right? So I show you some example that people have been doing, and it's very interesting to see that we can apply both chemicals and physical cues to trick the cells to become what we want them to be. So let's say this is the central role here, the pluripotent stem cell, IPS, or embryonic stem cells, and actually it could be the what is called adult stem cells. So anything start from here, and there's a lot of things going on, but I will pick one cell type that they want to direct them. Bear with me. Okay, so let's laser pointer. Okay, so let's say if you want to make chondrogenic cell, chondrocyte, cartilage. Yeah, this is what I working a lot on this cell type. So how are we gonna direct from stem cells to become this cell? There are so many options. For example, we can choose this pathway, one here and stop here first, and then do another set of cytokine, proteins, growth factors, and let them become the chondrogenic or cell types here. The second path could be this one. Go from the following steps here, move to the mesenchymal stem cell precursor, and then branching out to be this one, right? So ascorbic acid, TGF beta, so it's vitamin C, basically the red letters here, ascorbic acid, and transforming growth factor beta 3. So the name looks scary, but this is the components or the growth factor or the protein molecules that we add into the culture media to direct, differentiate stem cells to become the functional cells that we want at the end. So this could be the, let's say, the transit, like in the airplane. So we make these all the way and stop here first, and then we design what they want to be. It could be bone cells here, osteo cells here, or cartilage cells here. So you will see the branching point which change or modify the culture media, contain glycophosphate, dexamethasone, ascorbic acids, and become the bone here. And also this one doesn't have beta-vectazone, dexamethasones here. It's become cartilage. We take them out. Okay, so this one you can download and put into the Bibles onto your bedroom if you want to differentiate pluripotent stem cells to become any cell type. So you will see 4 different color code. This is mesoderm derivative, mesoderm cells, mesoderm, and ectoderms, which mostly are the brain cells, cortical neurons. Yeah, so, and this one, it will be the endoderm. So they use different set of growth media and culture. Endoderm, for example, the gut, the intestines here, beta cell, the pancreas cell, hepatocyte is here, and also germ cells to be zygote and sperm. So we can make things happen by change or modify the culture condition by using the biological cues and chemical cues in the culture media.
and they make something look like parody of the what is called table, periodic table. And rather than it's become the elements in this world, it's make up from the growth factors. So for example, activin A, one of the growth factors that we can add into the media, they have some details here. So for the cartilage, what we usually use, I make it in the green box here. So we add Win3A, GDNF, activin A, something like that. So in different times and amount, as I can show you here, from the left to the right. So the left side start from embryonic stem cells, human embryonic stem cells. So it's like a tiny little island, and it's not designed what they want to be yet, but we're gonna direct them slowly and become the nodules of chondrocyte-like morphology. Okay, so what they do is they add some growth factor, activin A, Win3A at the beginning, and then they stop it, and then add another set of growth factor, different amount. So the big one, the big letter, the capital letter here is a lot. And then they use a tiny little bit of NT4 during culture, and then they stop adding it and add polystatine instead. So yeah, so it's like I don't know, it's a good analogy to think about. We feed our dogs and cats different time, right? So the tiny little one, we add a different media, different recipes of the food. And then when they need more hair, shining hair, we add another supplement to make it grow. So the same thing pretty much when we do cell culture from embryonic stem cell to become the chondrocyte cells. Okay, this is physical cues and biological cues. And this one we apply, sorry, what did I just say? Chemical cues and biological cues. And this one will be more the physical. We apply the force, we apply the spinning during the cell culture. So you can see this one is called spinner flask, and it's increase the gas exchange. Rather than we let the cells sit stagnant into the culture plate, we spin them a little bit, you know, like, so it circulate the nutrients and gas transport. And another concern you will see here, there's some arrow which can generate the shear straight. Some cell doesn't like, they don't like pressure. They want to sit and stay in the bottom. So it depends on the cell type, so you can optimize the speed of the spinner flask. Another physical cues for the cell is called perfusion, and they have a great benefit as well, because you can see here when we see the cell, both red and green, into the scaffold, there are some nutrients which represent by the black dots everywhere, right? There's some part of the scaffold, it's basically in the middle where the nutrient cannot reach that much. The resulting of this kind of lack of flow, the cell inside the scaffold in the middle part can die because they cannot remove the waste or take the nutrients. So what the scientist doing is he generate the flow, which is represented by the arrow here. So the nutrients can reach the middle part of the culture system. Yeah, so there's more green at the middle, and this is the actual photo. Yeah, so in the center of the scaffold, there's some red dead cell represented by red color, and compared to another condition where the flow continuously, the cells happy, they are green and viable. This is the perfusion. So we can apply the what's called the pump and the flow into the culture system. Pretty much here. So this is the cartoon, and this is in the actual life. I show you here. There's the bones here, and there's a top part of the bone which is the cartilage. So we will see the porous. So the perfusion system will allow the media go through the porous structures of the tissue that we want to grow. Yeah, so the bone has the calcilates, which the pora like a sponge. What they did is there are 1, 2, 3, 4, 5, 6 channels where the bone plug can sit inside. I don't know if I should go to details of this photo, but yeah, for this one, so just focus on the right part of this photo, the bottom right. So we put the construct inside the channels, like a tiny little well, and there's a media, the red one, go up here, which basically can pass through the porous of the bone. Yeah, and then it will make like a box. It's like a tiny little box bioreactor. So yeah, the media can go out and recirculate to the main jar. And this is the breakdown into the schematic. So the scaffold of the tissue is here, and you will see there's some white channels can pass through. Yeah, so there's a media feed up and down, and there's some yeah, the holder here. Yeah, so this is the perfusion system where we can grow the bone tissue. Okay, and pretty much like a perfusion we call pulsatile, which very popular for growing the blood vessel. So imagine there's tiny little tubes here, which is the blood vessel, and they sense the pulse of the heartbeat. So what the scientists do, okay, there's a lot of going on here. Okay, so bear with me and we can go together. This red one here represent the vessel, and we zoom in, you can see here, and then yeah, like from top and both ends, they clamp with the holder, and this is the engineer vessel. We put it in here in the middle. Okay, so go back to the scheme. So this is the blood vessel, and there's a pump that feed the media through here, and this is the monitoring system. And where's the media come from? The media come from the reservoir, the red one here, and yeah, so it's go out here, and then the pump to feed the media. It's not continuous flow, but it's like pulsing. And you can see the pressure here. Pressure go up, they squeeze the media through the pump, through the tunnels, which is the vessel. Look like this. So everything happening in the bioreactor. And you on the left side of the slide, this is what it looked like. So you see the vessels in the bath, the medium bath, and it's clamped with the clips here, and there's some media circulates inside. So yeah, so pretty much like perfusion, but this is the pulse where have some up and down, like a like the heartbeats. Another thing that people using is the electrical signals, and they claim that it helps cell migration. Go to the black photo first. So this is a human umbilical epithelial cord. So it's epithelial cells from the umbilical cord. It stay like this. And here is the meniscus cells. Where are they? This in the หมอนรองกระดูก เขาเรียกว่าอะไร yeah, so it's one type of the cartilage tissue. It stays still on day 0. But when they apply the electrical stimuli, the meniscus cell, หมอนรองกระดูก cells, they can migrate towards the epithelial cell, another cell type. So the setup is look like this. So they have the carbon rods, the black one, 2, and one here, and another one's here, and they apply the electrical stimuli across the culture system. In 3 days, they can see it migrate faster compared to another one which not apply the electrical stimuli. The cell just stay where they are. But when we apply, yeah, they apply the electrical force, it shows something different. It stimulates cell migration into the wound. Let's say they try to close the gap, the black tiny little gaps here, start from big one and become smaller over time when applying the electrical stimuli. Another one that is very popular in cartilage tissue engineering, which is the mechanical loading. In real life, when animals or runners, they run on a treadmill, it showed that they had less chance to become osteoarthritis กระดูกเข่ากระดูกเสื่อมข้อเสื่อมนะคะ Yeah, so it's low risk when you do exercise and move, and they say that the mechanical loading maintain the homeostasis of the cells activities and increase the thickness of the cartilage and also produce more matrix. So exercise is good in humans and in animals in the lab. Yeah, so they have this concept and apply the mechanical loading into engineered cartilage tissue. So what they did is they make the cell-seeded agarose or cell-seeded scaffold. Agarose is hydrogel. I mean, put the cell inside the hydrogel or put the cells inside the porous scaffold, and they apply the force, just go up and down. You will see this picture is look like a plunger. Yeah, the white tiny and with the rods here. And also these are the engineered cartilage. So they put the hydrogels and the cell and make the tiny little disc. How many they have? Or maybe around 3, 6, 8. Yeah, they have 8 discs of engineered cartilage, and they close the lid with the plunger and go up and down. Yeah, so you see the directions of the movement. Yeah, so when they combine these two together, it looks like a tiny little box go up and down with a plunger, which simulate the walking or the running, so they can control the frequency. If it's go fast, it look like running. If it's slow, it look like walking. What they found is promote cell proliferation, which means the cell can divide it more, and they can the construct of the engineered cartilage produce more collagen and proteoglycan, which is extracellular matrix. And of course, the outcome is that the mechanical properties of engineered cartilage is very high and close to the native cartilage. Okay, so yeah, you will get some impression of physical stimuli, the mechanical loading, the electricity, the pulsatile, and the perfusion, right? And come to another aspect. Yeah, so it's easy if you want to apply the growth factors, the protein, the cytokine, the sugar, the drug. You just pipette or add these elements into the culture system to make the engineered tissue, to make a small piece of tissue, to make an organs or the organoid, right? And even you want to apply some physical loading, the compression, low oxygen, you just turn off the valves and the knobs. Yeah, that's it. It's easy. We can do it in the lab. And how about if we want to apply the microgravity in the culture system? What you're going to do? How you going to set up the experiment? Basically, microgravity is the weightless or almost no gravities. Yeah. How did you gonna set up this kind of experiment? Can you pipette or add the microgravity in the petri dish? Can you do that? Yeah. But the scientists in 90s, they have been done that. They managed to do it by go to the space. Yeah, so what they did is they bring the bioreactor and cartilage to the International Space Station. Back then they called Mir Space Station, but now they don't have a duty. So the Mir Space Station is not there anymore. They have International Space Station and Tiangong in China, but I know they have other countries have their space station up there. But this is what the scientists have been doing in 1997. I don't know anybody have already born. Yeah, so this is the first tissue engineering experiment in space on Mir Space Station, as I just mentioned it. I show you this example. It just gives you the idea that we can do it if we want to, and it's pretty cool experiment. Sorry. So what they've done is that they apply the cell, which is chondrocyte, into the scaffold, the porous scaffold, mix them together here, and then grow on Earth for 3 months. It's pretty long because maybe I think they probably make an appointment with NASA. Okay, this is the time, and you ready yet? And then when NASA come back, okay, it's time. We're gonna launch the space shuttles, bring the bioreactor and the cell to us. And then they have 2 groups. The first group, poor thing, stay on Earth for another 4 months with 1G gravity. Yeah. And another group, so lucky, they go to the space, which have low gravity. So they circulate here for 4 months and stay on Earth for a month. And if you take a look real quick, you will see there's some difference of the construct, cells and scaffold, right? So this one is stay basically in the bottoms of the bioreactor. I don't know why, maybe because of it's heavy, but here it's pretty much everywhere in the bioreactor. So, and the outcome is pretty impressive. Actually, cartilage be able to grow both in space and also on Earth. And you probably had the question why we care to grow the tissue on the space, right? Yeah. So because back then, and actually right now, maybe it's the things that we have to consider because maybe we can go and live on space or other planets soon, but our body is not designed to live at low gravity. Sometimes the astronaut needs to run on the treadmill and do some exercise because they're afraid of bone loss and cartilage damage, because up there are no force, there are no physical force, so we have to do a lot of exercise to resistance to build up the muscle and bone. Yeah, and then it could be another opportunity for us to design some experiments and apply the low gravities in the culture system, because maybe we can use it at some point. Okay, the outcome, what it look like. The tissue, the culture tissue on Earth is look like this. So they can secrete the extracellular matrix, which is the red one here. Pretty much the same on the Mir space station. But the things that I can detect here is the shape of the chondrocyte. Yeah, and you will see it too. On Earth, the cartilage is a little bit round, right? And then on Mir space station, it's the flat one, but they both function. Yeah, so you can find and read more about this paper in this topic. Yeah, in 1997, it's classic one for growing engineered tissue on space. And yeah, I think I keep the timing pretty good, so we finished before 3, and I touch on every topic here, cells and scaffold for your introductions, and then walk you through the experiment where people have been doing, and yeah, so both chemical cues and what else, and physical cue, mechanical loadings and everything. Last but not least, there are some rooms of improvement and there's some hope, because now the tissue engineer is already in the market. For example, 1997, 96, we have the skin graft called Integra already. And now 2006, there's some research and the actual application for engineering, tissue engineering bladder, use it. In 2008, the trachea is in the patient. So there's some hope, but you will see these are the structural support of the tissue. So the skin is not that complex. The bladder is just the shapes and put the cells in there, and also the trachea is look like a tunnel. But I haven't seen, maybe you've seen it. How about the heart, tissue engineer heart, like the whole heart? It's so challenging. I don't want to say negative thing like we cannot do it, but I think there's some room of improvement to reach that point. I haven't seen the heart. I haven't seen the kidney. The kidney, the doctor told me that it's very complicated. It's very challenging. Heart, kidneys, pancreas, and liver could be the potential to do it So in my opinion, but we have to work on the enzymes, timing, how when the enzyme will come out, yeah, something like that. So it's totally a different stories when we think about the structural support and the tissue that secret things. Yeah. Okay. Last but not least, if you want to create the engineered tissue, I suggest you to talk to the doctor, because it's the collaborative work. So since now everybody's in the room there, we are engineer, we know how to build things, optimize, we can control the degradation. So basically we on this side of the research, we are mechanical engineer, nanotechnology, like we material science, we know how to combine things together, but maybe we don't know the needs of the patients or the doctors. So this is the example of dermal regeneration templates, where the mechanical engineer meet the surgeons, because the surgeon has the need to develop the wound healing patch. So they talk and work together and create the integrated artificial skin, which basically there's no cells in there. This version, the original versions. So it's like the membrane and put into the wound size and let the cell, the patient tissue and patient cell migrate through the collagen sponge here. And on the top, there's some silicone patch to prevent the infections and liquid blood fluid lost. So, and then they're gonna, when the wound almost healed, they remove the silicone out and it's perfectly healed and prevent the scar formation. So it's very important when you develop some kind of engineered tissue, talk to someone who know anatomy. Yep. Yeah, this is the key message that I want to talk to you guys. So clinician, they close to the patient, they have the medical questions. But for us, we know how to develop the device and research discovery, we construct things, but we have to work together. So yeah, that is it. Ajahn Solada, I think my class is like this. And I don't know if you want to summary, if you, I don't know how things work, maybe is it possible that you have your question in your mind? For example, you want to make the brain tissue and how you're gonna incorporate the triads. Choose the cells that you want and choose the material that you think is good, and then what kind of growth factor to stimulate the brain development. Maybe it's too much for the time because you have to submit by today. Yeah, so we can discuss and minimize the task rather than
it could be the principle of making tissues.
Yeah.
What do you want and what do you have to control, something like that. I think whatever, because you will have a chance to read over Ajahn Sukhansa, to see the impression of student on your on your lecture. So questions, anyone in this one? I think this one you are talking about signals and emphasize on physical signals, right?
Right.
And actually we have a lot of signals in the tissue engineering. Once we made tissue engineering, actually students should know that we did tissue engineering in vitro, means not inside the living, and also in vivo. And sometimes we use both. We kind of culture outside of the living and then move it inside the living also. So there's many kinds of signal that you can give it to get to control the tissue formation. So Ajahn Sukhansa, did you talk anything about chemical? Can you comment on chemical signals also for the tissue engineering?
Yes. Oh, I start at the very beginning.
Okay.
Like this, it's like periodic.
All right.
Yeah. So
okay,
it's easy to apply. You just follow the protocol. When to
okay.
stop, when to start.
So
Yeah.
because cells want to eat something, it's like nutrient for metabolize, right?
Yeah.
But so they have chemical signal then you can find the formulation from the database, what kind of cell want what kind of nutrient or what kind of chemical stimulation. Okay, but Ajahn Supansa talked a lot about physical stimulation. For example, mechanical stimulation. Okay, and did you talk about temperature at all? Not sure. I'm not sure that temperature will do anything because we never heard of this much. Mechanical stimulation, electrical stimulation. Okay, I heard of some ultrasonic stimulation, right? In cell culture, I'm not sure.
And
Ultrasonic,
the oxygen
right?
level too. Oxygen
Yeah,
level.
oxygen signal, because we thought that we need a lot of oxygen, but it's not, maybe it's not the case, right? Ajahn, can you comment about oxygen a little bit? Because the reason I asked, because student had learned at least 3 times on bioreactor, so they kind of very concentrate on this bioreactor. Yes, you can go into oxygen control, please.
Right. For me, it's I don't know, it's I try to work on this hypoxia, which is low oxygen a lot. Yeah. Let's start from this one. So we breathe here, everybody is in this room. We encounter, we facing 21% oxygen at the air, at the atmosphere. And when we breathe the air inside the lung, it's dropped to 13%, 10%, 21, and then 10. And inside our capillary and the lung, it could be only 5%. And in our fingertips, it could be 1%, or in the joint of the cartilage, there's no blood supply at all. It's basically our body inside, deep down, we're facing hypoxic, low oxygen tension. But in the lab, we let the cell grow in the 21% oxygen. Maybe it's kind of not physiological condition in the lab, but in our body, we need very little. So that's why the trend of hypoxic or low oxygen tension to culture the cell inside the lab, it's now in the study, in the research.
Yeah,
scheme,
interesting.
right? Yeah.
I think the one of the things that we culture as the bioreactor for tissue engineering, I think it's different from the bioreactor that students used before. Okay, it's very interesting topic actually, because the student use liquid state bioreactor, liquid state, gas state bioreactor to make more oxygen to get inside. But to make tissue is actually mostly solid state or hydrogel state, mostly, because they don't have that much water, but they have the the hydrogel status or something like that, bioreactor. And another thing is sometimes they use buffer environment is very important, because pH has to be stable for the cells. So buffer state, that means you have to have carbonic buffer and using carbon dioxide environment, something like that. And if you heard of carbon dioxide incubator that we use for cell culture, so it's kind of different way of doing bioreactor from what you guys have seen before. Okay, so questions, anyone? Okay, you think you can finish the 1-page summary within tonight by midnight? If yes, I have to thank you, Ajahn Supansa, because we have inside information and very exciting slides a lot, the tissue formation, tissue engineering, something like that. Even I didn't do, lecture on tissue engineering for almost 10 years already. So I kind of enjoy your teaching, and I hope next year you will become instead of our invite speaker, will become the regular speaker, and then students can learn more on tissue engineering topics from you. Okay.
Yeah, just give you the flavor of each component.
Okay.
So I didn't go to the detail because
And also student, this is the last lecture on introduction to bioengineer, you can see that bioengineer is a large word. Many things, including cells and the living, that you can apply engineering principle into it. It's all considered bioengineering. Very strange, but yeah, it's true. So you might feel overwhelming, but I hope you know how to read them in the future, because you have to self-study most of the time. So we touch on lightly and very wide and general on each topic. I would like to thank you, Ajahn Supansa, very much. And okay, I think if nobody asks questions.
Actually, there's someone on a chat.
Oh, please, thank you.
Can you explain again why we have to do tissue of cartilage experiment in the space? Yep. So it's because back then, originally there are some Soviets and America, right? So they want to be the pioneer of space. Anyway, yeah, so they both managed to go up there, but the core question why we want to answer want to do experiment up there, because yeah, the astronauts again, yeah, so they live there and then they want to pioneer and stay a little bit longer. It's more like a science fiction and competition between the two powerful countries. So but they care more about the life of the astronaut as well, which can represent the humankind of us. Maybe some of you guys can go up there and stay a little bit longer without the muscle loss or bone tissue loss or cartilage defect. So it's like, and they use cartilage as the representative of tissue to grow up there and see the effect of, life in the space. And yeah, so pretty much they want to see how can we survive in any kind of low microgravity, the microgravity effect on the human body. So basically it's a curiosity, and will develop and the new methods to make us survive up there longer without the severity of muscle loss. Yeah, something like that. That's why they invest a lot of money to grow things up there. You will see not only the tissue, they grow some basil, they do a lot of I hear there's some synthetic pearls up there as well. So maybe we can move to space at some point very soon. Yep.
Okay, ka. Anyone want to ask anything more? Oh, if not, I would like to thank you, Ajahn Supansa. I think you have you can have a contact with Ajahn Supansa, because Ajahn Supansa use Facebook, even though
I don't know. I don't